So a little cavity here where we can put a germinating seed in, and then a flow channel to pump by the nutrients, but most importantly for this set of experiments, change the flow rate that these nutrients go by, because that changes the pressure that's applied to the developing root. And what they were studying in this work was to see how that changed the root morphology. So here we can see that the little plant was germinating, growing, and the root would slowly find itself a way down the microchannel. Here you can see that with different flow rates, the length of the roots changed, but there was an optimum at 0.4 microliters per minute. It was a relatively long root being formed, but beyond that maximum flow rate, actually the root length became shorter again. So here we can see the quantitative analysis that was done on this developing root, where at different time points the root length was measured for the different flow rates. And over the different intervals we could see that, so here are the lower flow rates, here are the higher flow rates, and we can see, as we already could see on the diagram here, there is an optimum where we get the longest roots being formed, after which the length of the roots gets smaller again. So here we've got an upwards trend with flow rate, here we've got a downwards trend with flow rate. After taking these roots out of the microfluidic devices and studying them, it became obvious that if we increase the flow rate, the number of cells separating the core of the root from the shell actually reduced. So at a low flow rate we had a relatively large space here, and this area, the thickness of this area changed depending on the flow rate. And here we've got a way of looking at the pressure that is being applied. So the loading flow we can calculate with the flow rate, what actually the pressure is that is applied to this plant root at the interface. And we can then dissect the roots. We can see that the size of the roots, the outer diameter, actually didn't change with the flow rate. So we, as a result of the different flow conditions, this research found that whereas the exterior of the roots looked very similar in diameter, the cell differentiation inside these structures was very different, and we got changes in the core to phloem ratio depending on what the pressures were that were applied to the outside of the root. And that's something no one had ever seen before. There have been no other tools where we can apply a continuous pressure to a developing plant root to actually look at the morphological changes of the different cell types, the differentiation of the different cell types inside a system, and then take them out and allow them for analysis by microscopy. So the changes in cell differentiation and therefore the morphology of root as a result of different pressures is called thiomorphogenesis, and this was studied in using a microfluidic device. The important findings coming out of this research was that increasing flow rates boost the root length and the nitrogen uptake until reaching an optimum flow rate. At too high a flow rates, there's clear stress on the cells, and the roots don't get any longer. Moreover, due to the high stress, they get even shorter. They get shorter again. The roots in the flow traditionally consistently outperformed those in no flow conditions. They grew faster because they were able to take more nutrients out of the flow because there was more nitrogen flowing by. And nutrient flow induces the morphological changes, offering new insights in root adaptation and cellular responses to changes to the physical environments. So very simple microfluidic device, but by understanding the fluidic properties, by understanding how a flow changes the pressure, a whole range of new experiments could be developed. After the experiments, the roots could be taken out of the device and characterized using microscopy, leading to new biological understanding, which in turn potentially can be used for hydroponic culture and understanding how to optimize flow rates in hydroponic culture in order to get better crops, get good strong root development, but not to get to the flow rates that get to too high a shear stress that again then lead to decreased root development. So using microfluidics can be used in agriculture to get new understanding to then enhance the microscale phenomena again. Looking at an other option is to look in a closer detail at nutrient uptake. And here we've got a microfluidic device where again the developing plant can be studied. And what this device has is that it's got two embedded electrochemical sensors. One of these sensors has got a functional layer that it measures phosphate, the other one measures nitrate from memory. And the picture in the set of slides that you get, the picture is a little bit better than here on the screen. But we've got a little fluidic flow cell here with two different nutrient sensors, each selectively measuring one analyte. So over time we get time-based profiles for two different analytes. And we've got a little cradle here to a little germination pot where we can add the legume seed that is with growth going to be consuming more of the nutrients that we can then monitor. So this is the way that the different electrochemical cells were created, where we've got a working electrode, a reference electrode, and they were functionalized with materials to create selective electrodes for nitrate and phosphate detection. So here we've got the two different electrochemical sensors. We've got the little seed being placed there. The seed develops a root and starts taking up nutrients out of this containment. We can see that the distance here, the different measurements are being taken on different days. And we can see a very quick decline in our nitrogen concentration compared to the phosphate. At day 5, we get fresh solution being introduced. But we can see that the slope here in the nitrogen uptake has increased compared to the early time, which is not a weird observation because we've got a much larger plant after that 5 days. And therefore we've got a larger metabolic capacity, which correlates with a larger capacity to take up nitrogen. So this allows, in this instance it was made for two obvious nutrients, nitrate and phosphate, each having their different rate of uptake. But by changing the chemistry at the sensor head, you could change the nutrients that you're looking at. And you could, for example, start studying the role of certain micronutrients at the development of stronger root systems, or the rate at which we start getting to the 2-leaf, 4-leaf stage. So by being able to monitor the nutrient uptake, we actually know as a unit of time at what point in time the developing plants need what nutrient profile. So another part in agriculture is not necessarily looking at the development of plants themselves, but look at the development of disease. And this article here is the microfluidic platform that was used to study microbial spore germination under different conditions. Here we've got some picture of the microfluidic device, and there's two fluidic features that are important where we take advantage of microfluidics and miniaturization. The first thing is that because our microfluidics allows fine scale control, it allows us for the loading and the distribution of different bacterial species into a device and study them individually. So we can create little reservoirs, little cavities, where each of these spores can be individually placed, and using a microscope we can monitor the germination of multiple spores in parallel. But we don't look at an overall spore development, we don't look at averages, we can look at the development and germination of individual spores. The other microfluidic feature we can use is the fact that we have laminar flow. Because if we've got laminar flow, we can create and control concentration gradients. Here on the right-hand side, we can see that we have some high concentration fluid coming in at this side, some low concentration of our inlet coming in, and we can see a stepwise increase of our chemical, in this case fluorescein, because it makes it nice and easy to visualize, and that will continue through our flow cell. So if we have our little tray loaded full with spores, so every spore has got its own little cage to be sitting in, we then know that all the spores that are sitting in this area are not exposed to a certain reagent. All the ones that are sitting here are at about a quarter concentration, 50, 75%, 100% of concentration. So we have a concentration gradient, and we can conduct cell-based studies on individual spores under a range of different experimental conditions without having to really do anything. There's a microfluidic structure that allows you to make this concentration gradient. So you just introduce water and your reagent that you're looking at. The fluidics will take care of the fact that we get this gradient formed. This gradient will flow over these different bacterial spores, and we can, using a microscope, monitor them individually, do large-scale image analysis, and do a number of experiments that usually would have taken weeks in the lab in a few days by just doing them in parallel. Here we can see one example where we see the germination of one of those spores with Bacillus subtilis, and you can see that over a 2.5 hour period you can actually see the morphological changes as a result. So what they did...
Can you go to the back slide? I couldn't get that, how the microfluidic device is giving us the...
It is an area where you split the flow and split the flow again. So if you've got 100% water and 100% fluorescein coming together, and you then split the flow in two, then you get 50-50 concentration. If you then add more water coming in, I'll share another paper. So you can use the flow resistances of different networks to have different mixers and create a fluidic gradient using flow resistance. I'll show you one of the devices. It's one of the things that's impossible to explain. And once you see it happen, it works like a Christmas tree. So it branches out.
Okay.
You get different ratios of water and dye being mixed.
You haven't got your box yet?
No.
Oh.
Otherwise I would have shown it here. So here we've got the germination rate at a nutrient broth, a synthetic media, and L and D alanine. And what they looked at was the germination of the different seeds. You can see that in the nutrient broth we got very quick and good germination. L-alanine was clearly important there because we still get, when only alanine was there, we can get good germination. If we get D-alanine, there is no seeds germinating, so that's not good for the development of bacteria, or excellent if we don't want the germination to take place of these bacteria. And if we look at the synthetic media, looking here at 46 different in parallel, they just don't germinate. So there was some statistical analysis being done on how the concentration of the alanine affected the germination rate, realizing that you got really good germination at a higher concentration, and at a lower we actually had a reasonable fraction of them not germinating. No significant differences were observed between the 1 molar and the 100 micromolar, but the spread for the 1 molar, the spread in germination rates was slightly higher, and therefore the 100 micromolar was picked as the optimum to get synchronized and fast germination of these microbes. So here you can see a little movie where you can see the morphological changes happening over time of one of the bacteria that was studied. And this is again this is done with a microscope and with so many different experiments happening with microscopy. Image analysis actually allows you to track things based on location and time, and then you can quite quickly analyze the data and see what's happening. So here again we've got concentration changes of one of the other reagents. Yes, so it's indeed not the reagent, it's the no, it is different concentrations. I just don't know what reagent it is, and it's not here in the note so as well. But again, we can see that we've got changes in germination time depending on the concentration, and at the highest concentration we got no germination at all. So our optimum condition, if you wanted to have germination, was to be at the lower concentration range. So the reason for putting this in, it shows that you can actually look at individual bacteria. If your problem with bacteria formation is that you don't want the spores to germinate, this allows you to test a whole range of chemical conditions where you do or you don't get germination, which provides you a very good starting point of working with selecting chemical conditions that you can prevent the germination of these bacteria, unless they're ones that are used for fermentation processes. So if you want to promote, you can also select those conditions. So early detections of threats and disease is one of the parts that I like to work at. And of course, in agriculture, there's a whole range of different microorganisms, bacteria, fungi, viruses. There's a whole range of different microorganisms that can affect the quality of vegetables, fruits and grains. And recently there was a really nice review on how microfluidics has helped with the advancing the early detection and the on-site detection of all these nasties in the agricultural sector. One of the ways that can be helped is using Internet of Things. So have little microfabricated devices in there with a sensor that will be able to give you a reading that transposes on your phone. So you actually don't need to be on site to do the monitoring. So they made a device using a dry film photoresist. They used photolithography, so they used a mask to selectively make part of that photoresist soluble, part of that photoresist insoluble, and create a fluidic structure that way. This allowed them to make a negative mold, to then cast PDMS over to create microfluidic channels. And they then sealed the microfluidic channels together onto a glass slide to go from an open channel to a closed fluidic device. What they also did here was to cut a little hole in an acrylic device, because in the ultimate device they wanted to have a little reservoir where they could do spectrophotometric measurements. The whole device was relying on having your sample coming in with a reagent, having a microfluidic mixer to allow the target analyte to actually react with the reagent, and then measure the absorption by putting an LED with a fixed wavelength on one side of the detection chamber, a photodiode to the other side, then use an impedance amplifier, a single board computer, in order to either through Bluetooth transmit the signal to a laptop or a phone, or to use an Internet of Things module to directly get the data stored in the cloud, so that people could, from a very different location, understand what the, in this case, the nitrate concentrations were. With the reagent, the method was validated that there was an increase in absorbance with the increase in concentration of a target analyte from 1 molar to 1 micromolar to 30 micromolar, and it was also confirmed that with increasing concentration, the detection voltage coming out of the photodetector decreased. So the change in signal could be correlated to the concentration present, and therefore the system could be used for the monitoring of the nitrate. This is some of the work that we've done probably 10-15 years ago to see if we could decrease the sample preparations of fruits and vegetables for mineral analysis. We had a project, and the students, we really were struggling to get bits of fruit chopped up, see if we had to freeze them, grind them with a mortar and pestle. And our question is, do we have to go through all this work, or could we just be lazy? And could we put a bit of fruit in our analytical instrument and inject our analytes? So here we've got a bit of zucchini being stuck in a sample vial. In this instance it was a capillary electrophoresis instrument, but it's exactly the same types of vials that you load into an autosampler for a gas chromatography instrument or for an HPLC. We found to our great delight that indeed we got a nice profile, nice separation of our inorganic cations when we injected directly from a bit of zucchini, but we had horrendous repeatability. We had no consistency if we had a bit of the same zucchini. We got very different results, and initially we blamed biological variability because that's always a good one to blame. But we thought that that was a little bit too easy. And we then decided or realized it was actually not biological variability, it was fluidics taking place. Different plant cells carry different amounts of water, and that actually applies a fair bit of pressure. So if you poke a few silica capillary, a glass capillary into glass, sometimes you're in the interstitial space, sometimes you are in a cell itself. The pressure that's being applied is actually different every injection. If you apply a different pressure, as microfluidics people, we should have understood this. Different pressure means different volume. This means that if you randomly poke a capillary into a bit of zucchini, sometimes you are in an area where you've got high pressure, sometimes in your area that you've got low pressure. You inject different volumes, which means that your calibration curve doesn't work anymore. We decided to fight back, and we're like, how can you fight back against pressure? You just want to apply more pressure from the other end. We didn't quite want to apply mechanical pressure, but we introduced the viscous reagents. Viscosity increases the back pressure, which means that by increasing the viscosity inside our capillary, the differences in pressure from where we injected the capillary into the zucchini actually didn't matter anymore. So increasing the viscosity means that we needed a much higher pressure to get a significant variability in volume introduced in our capillary, which means that we stop the zucchini's ability from squirting liquid into our fused silica capillary by just having a highly viscous solution in there. If you then apply a potential difference, you are doing a purely electrokinetic injection, which then means that you get good repeatability. And as you can see here, we've done the analysis of potassium, calcium, magnesium in zucchini, apple, and mushroom. We also did tomato, strawberry. We just got the whole vegetable shop and just put them all in different vials. We got the separation to work the same way. And we confirmed the analytical answers that we got out of our capillary electrophoresis method with ICP-MS. And we got a good correlation between the two different results. So we could cut down a significant amount of sample preparation here by directly injecting our analytes from the fruit. We could only do this with some understanding of the fluidics, because when you put the capillary directly in the fruit, the pressure, depending on where you end up in the cellular structure, the liquid pressure generated from there is very different. So with an aqueous electrolyte, without any viscous reagents, the pressure differences between the different locations in the fruit made a significant contribution, and therefore we had poor repeatability. But if we increased the viscosity, the pressure changes became negligible because they couldn't overcome the back pressure of the liquid inside our fused silica capillary anymore, and we could get consistent results. Another example of applications in agriculture is to look at the detection of fungal infection biomarkers. In this instance, there are 3 different fungi, I think they were. 3 different pathogens that could affect grapes. And if the grapes were being impacted, there were increased concentration of jasmonic acid, azelaic acid, and salicylic acid present. So the idea was to develop a device that these 3 different biomarkers for fungal infection could be detected at the same time. So here we've got the microfluidic device that was again integrated into a system onto a printed circuit board, but here we've got the fluidic device itself, where we have one side where we can introduce the sample, and we've got 3 different analytical zones where different nanoparticles were created to conduct 3 different assays. So we've got one sample coming in. We've got three different areas where 3 different assays can be taken in parallel. And then using the photometric results, we can quantify the concentration of these 3 different biomarkers, and with this assess if our grapes have been affected by one of these fungi or not. The first reagent that was used were some functionalized titanium dioxide nanoparticles that led, in presence of the salicylic acid, led to some agglomeration, whereas in absence they didn't. And we could get a linear concentration of the optical signal based on the presence of the absence of the analyte. The second assay taking place was the conversion of, was an enzymatic assay that was being conducted. So there was some tyrosinase and its substrate, and in presence of our biomarker, so in absence of the biomarker, the enzyme would convert all the substrate, whereas in presence of the biomarker, we got inhibition of the enzyme, and therefore we got an increased presence of the substrate, which then could be connected with our enzyme-based assay. And again, good linearity of the concentration of the biomarker relative to the response. In the first instance, we've got an ELISA, an immunoassay, where we get the binding of our biomarker to protein and then to an antibody for detection. So again, in absence, we have no binding of our biomarker to the BSA and therefore to our antibody, and we have no signal. Whereas in presence of our analyte, we do get our sandwich assay to work, and a complete ELISA assay could be conducted for this biomarker. So in this article, the 3 different assays are validated, and it allowed for multiplex detection of the 3 different biomarkers through the development of 3 separate selective assays. This in here is a work where we used a pumpless acoustofluidic device for rapid detection of pathogens in fruit. And again, we've seen this device come by past. What we used was very quick mixing of these acoustic micromixes by having the triangular structures next to a bubble, creating these curvature flows that we've just already seen come by with the nanoparticle synthesis. In this instance, the micromixers were used to homogenize the fruit mixture with the lysine solution. Then there was a filtering functionality at the end to stop any cellular debris. And we could, with a pipette tip, isolate the clean sample from the other side. The liquid flow, the fluidic flow in this microfluidic device was driven by a highly wicking fiber. So we didn't need any external pumps that needed electricity. We just put a highly hydrophilic fiber that we extracted from nappies. We put them at the outlet of the microfluidic device. They wicked up the liquid, acting as a pump, drawing new liquid through our micromixer. The little speaker that we'd extracted from a speaker that we bought somewhere in a cheap shop. A speaker allowed for the quick mixing, and the thread-based device here allowed for pulling the liquid of the heterogeneous strawberry or raspberry extract from one side to make it homogeneous at the other and ready for amplification of the DNA. So we looked at the mixing efficiency under different frequencies, and we could find an increase in DNA copy number by PCR if we spiked more E. coli onto the fruit at the start of the assay. So we started off with a piece of raspberry, mimicked the contamination by depositing a known amount of E. coli, and we could indeed use the homogenize the fruit as well as mix the fruit and the bacterial sample with lytic reagents, use the thread to drive the liquid through the microfluidic device, and then the inbuilt filter stopped all the cell debris from coming to the outlet, and then we pipetted that sample into the PCR and amplified the DNA in order to allow us to quantify the number of DNA that we found back on the sample. The last device I think I want to talk about is a simple paper-based device for the detection of some parathyroids in environmental water samples. So that is, the parathyroids are a group of pesticides that are frequently used in agriculture, but you probably don't want to have on your tomatoes. A coulometric reaction that was selected for these reagents was developed. With that came a microfluidic device. If you remember us playing in the lab with the paper microfluidics allowed us for the creation of hydrophobic barriers where the aqueous solutions and reagents didn't want to go, and leave some hydrophilic patches where we could transport our reagents and add our chemistries. So here we have some paper impregnated with paraffin oil, so acting as a hydrophobic barrier other than in the areas where we're open. We've got a nihedrin layer where little discs soaked in reagents. We've got a buffer PVP layer where we've got little discs with that reagent. On top we've got a laminating pouch that's transparent but has some excess hole to deposit the reagents. All the way on the bottom of this device we have a transparent film that is not allowing any of our aqueous reagents to go through, but provides a leakage tight barrier so that the reaction can actually take place. If this is all put together, we've got a sandwiched device that's got a water impermeable bottom. It's got some reservoirs defined by using hydrophobic barriers. It's got discs that provide different reagents, and it's got excess hole that we can pipette different samples into. And indeed, we selected the coulometric reaction to be in there, and depending on the concentration of each of our 4 target pesticides, we got an increase in color intensity of the coulometric reaction on the paper with increasing concentration. And luckily, with other known pesticides that were not part of this class, this coulometric reaction didn't take place. So we could quantify based on the change in color. The people here were able to quantify the amount of this class of pesticides present, knowing that the other commonly used pesticides could not be detected by the same assay. So that wasn't the last one. This is one looking at acetylcholine esterase inhibitors. Again, very good pesticides. Not very good if you're a farmer, you're exposed to them, because then most of your own pharmacology doesn't really work anymore. Here we've got a droplet-based reaction system where we've got the acetylcholine esterase, which is an enzyme that's present in our bodies for the breakdown of neurotransmitters. In this instance, we wanted to know the presence of inhibitors to this enzyme, because they are toxic for our bodies but also toxic for some pests, so they're frequently used as pesticides on the farm. If we've got the inhibitor absent, our acetylcholine esterase happily converts acetylcholine into its esters, which can then be oxidized and measured electrochemically. If we've got an inhibitor present, we inhibit the activity of this enzyme, which means that we lower the activity of our acetylcholine esterase. We've got less of our thiacholine being formed, which means that we got less of our oxidation reaction, which means with an electrochemical reactor, we get a lower signal. In this instance, the assay was conducted into little droplets. So here we've got oil as our continuous phase. We get our aqueous reagents, which are our enzyme, our substrate, and our sample to come together in a little self-contained system in our microfluidic droplets. Mixing will take place automatically because we get the circulatory movement happening inside our droplets. Then our enzyme can get to work. If our enzyme is working fine, we get a high signal from the electrochemical detector. So you can see the working and the counter electrode being introduced here with the reference sitting elsewhere. So we get a nice high signal if our enzyme activity is high. However, if we had an inhibitor present in our sample, we get a decrease of signal measured by our on-chip electrochemical detector. Here we can see some electrochemical characterization of our detectors, and we can see that we can get an increased detection signal with an increase in analytes. And here we can see our droplet passing by our detector. If both of our detection electrodes are in contact in our aqueous droplets, we get electrochemical conversion. If we're back into the oil phase coming by, we get no electrochemical conversion. So we can see each of our droplets coming by because our electrochemical detector only provides a signal if it is in contact with the electrochemically active species. Using the flow ratio, so you can see that we had the 3 different channels here. Using the flow ratios between our substrate and our enzyme concentration, we can add different amounts of substrate. So here you can see changes in pink color, pink intensity in our droplets. So we can potentially change our substrate concentration to do more advanced enzyme kinetic assays in the device. And here we can see a series of droplets coming by. If an inhibitor is injected, we can see that if there's no inhibitor present yet, we get high currents because we get a high signal from our electrochemical conversion. If we have a high concentration of an inhibitor present, our esterase doesn't work anymore. We can't do our electrochemical conversion. And if we stop introducing our inhibitor, our signal recovers, and we can understand how much of our inhibitor we had present based on how much our signal intensity drops here between having no inhibitor present and having our inhibitor there. So you can see here in the lower signal, we've got more signal left than we had under A. So as a summary, in agriculture, it's a big field. There's many applications. Some of the applications study the biological processes, which could be optimizing nutrients. It could also be looking at how different force fields affect plant roots. We've got analytical applications, which could be detecting nutrient uptake, optimized nutrient profiles. It could be detecting pests, as well as the pesticides to ensure the safety of consumption of the reagents. Additionally, we can create nanoparticles for agriculture production that then facilitate growth. That would be an application in agriculture as well. I just saw my phone buzzing that it's time to go for lunch, so I'd suggest we start 15 minutes later as well. It's time to go and eat something, and I'll see you back for 1 hour of lectures and 1 hour of a little bit of an idea how we'll do the exam later on. Thank you very much for being patient.
You must have tired as well, speaking 3 hours.
It's the job. It's part of the job here.
But I must say, you have a good